Good morning in Colombia, good afternoon in the United States. I am Angela Amarillo, the director of the doctoral program in Environmental and Rural Studies at the Pontificia Universidad Javeriana, Colombia. Welcome to our conversations in environmental, ecological, and rural studies. Today, I have the great pleasure to introduce two extraordinary researchers, Dr. Rosemary Grant and Dr. Peter Grant. Dr. Rosemary Grant is an emeritus and senior research biologist, and Dr. Peter Grant is an emeritus professor of zoology, both of them at Princeton University. For over 50 years, Dr. Rosemary and Dr. Peter Grant have dedicated their research to the fascinating world of evolutionary ecology and evolutionary biology, for which they have worked with Darwin's finches on the Galapagos Islands. Their research has also been the subject of many of our courses and has inspired thousands of young scientists to pursue the path we share today in environmental and rural and ecological studies. Their groundbreaking work combines ecology, behavior, and genetics to understand the intricacy of the interaction between ecology and evolution and how this relationship shapes biodiversity. They will be presenting the talk from microcosmos to microcosms, adaptive radiation in Darwin finches. Dr. Rosemary and Dr. Peter Grant, welcome to the doctorate program in Environmental and Rural Studies at Pontificia Universidad Javeriana. We are very honored to have you both join us today. With no further to say, we are now ready to listen to your talk. Thank you very much, and the audience is all yours.
Thank you very much for that very kind introduction. Oh, share screen, just 1 second.
Share.
Share. There.
There we go.
There. Is that, can you see that? Yeah.
We're hoping you
Yes,
can.
we can see. Thank you.
Yes.
Yeah.
Okay. Thank you very much.
Right. Well, thank you very much for that lovely introduction, and also for the invitation to come into this room and tell you about Darwin's finches and our studies. One of the greatest challenges that evolutionary biologists have these days is to explain the enormous diversity on this planet. The rich biodiversity amounts to millions of species, and there are so many, in fact, that we do not know to the nearest 1 million how many species of organisms there are, which I think is truly remarkable in these modern times. Consider, for example, this little group of fish. It's a small group of cichlids from Lake Tanganyika in Africa. In fact, it's part of a group of more than 100, in fact, hundreds of species, all derived from a common ancestor. And it is known as an example of adaptive radiation from an ancestor radiating in divergent groups and directions. Now, here is another one from the Caribbean, Anolis lizards in about 135 species in total, and once again, all derived from a common ancestor, or a very small number of ancestral species. And the question they pose, as well as the bigger question, is how do we explain this enormous diversity? Well, for any group of organisms, there are 4 questions that we would like to be able to answer. We would like to be able to establish the history of the group, the environments in which they diversified, the traits that are important in the diversification, and the genes and gene combinations that underlay the diversification. Our chosen group is a group of 18 species of Darwin's finches on the Galapagos Islands. It's a young radiation, that is, the radiation from a single ancestral species took place in about 1 million years, or perhaps a little bit more. Much of the Galapagos Islands is in its pristine state. And so for that reason, and for the fact that the environment is very dynamic, it's a convenient group for studying evolution as a process. Notice that the traits in question are body size and beak size, not color, but beak size principally and shape. And the beak is a tool for exploiting the environment. So the radiation is an ecological radiation permitted by the remoteness of the environment, and therefore the absence or scarcity of other species with which, excuse me, they share the environment. Well, they are volcanic in origin. The islands are volcanic in origin, and most of them owe their origin to hotspot activities below currently south of Fernandina Island. You have to think of the islands as being large lumps of magma that are heading towards the mainland on this geophysical plate at the rate of 60 kilometers per million years. A very dynamic environment. I have frozen up for the moment.
Try this.
Excuse us a moment while
Try this.
while we undo this.
Yeah.
That's better.
Yeah.
All right. And not only is it a geophysically highly dynamic environment, but also in terms of climate, the islands have experienced 10 cycles of alternating glacial and interglacial cold and hot conditions, which has affected the sea level. And as a result, the islands at times have become closer and higher than they are at the moment. And in fact, at the extreme, when the sea level is lowest, some of these islands have coalesced when previously separated. So in this dynamic environment, how do we explain the origin of and diversification of the finches? Here is the macroevolutionary pattern rendered in molecular genetic terms in the form of a phylogeny dated to about 1 million years. That is the pattern for which a microevolution explanation is required. The key factor that we should try to understand is how one species gives rise to two, because when we've got the answer to that one, we can multiply that answer up many times to account for the total radiation. Now, in essence, speciation is the splitting of a single lineage into two through two populations at the minimum being formed from one, and diverging to the point where the lineage is so formed no longer can interbreed, in which case everybody would agree two species have been formed from one. Darwin had a clear idea of how this process took place. He believed that spatial separation of these two lineages in different locations would enhance the process of divergence. Even in the absence of any understanding of genetics, it would allow that to happen. And here in simplified form is the classical explanation for how two species are formed from one. Classical in the sense that the essence goes back to Darwin. And it involves 3 steps. One starting with the arrival into this new environment of colonists, in this case from the South American mainland, further spread of populations, establishing new populations on different islands through dispersal, and then the coming together of previously separated populations, already different, having acquired adaptations and differences due to random genetic drift as well, in separate locations or allopatry. And then in sympatry, where they come together, they may be expected to diverge further as a result of natural selection that minimizes 2 things. One is interbreeding, if the offspring of such breeding are relatively unfit. And secondly, competition for resources, principally food. Now, that second one is understood by the term of character displacement. And I will give an example later from the studies that I'm now about to describe on this little island in the center of the archipelago called Daphne Major. It is, in some ways, an ideal setting for understanding microevolutionary processes that can give enlightenment as to how the macroevolutionary pattern of many species in the radiation unfolded. It is 120 meters high. It is about 3/4 of a kilometer long. And in the middle, there is a crater. Now, first of all, it is occupied by 4 species in the genus Geospiza, or ground finches. There's a large ground finch, a medium ground finch, and a small ground finch, and then a cactus finch with a relatively elongated beak. As I said, the island has a crater in the middle. Birds fly everywhere. We follow them by also going everywhere. The birds nest in the bushes on the flanks of the crater, and that's where we concentrate a lot of our activity. So we sleep in a tent. Excuse me just a moment, I'm just checking on one thing. Okay, we sleep in a tent, and we cook in a cave, which doubles up as a laboratory. And like many people who study birds, we put up nets to capture them and then take them on this little hot island into a relatively shady period where Rosemary takes a small drop of blood and stores it for transport back to North America and extraction of DNA for further genomic analysis. We'll come to that later. But my role in this part of the process is weighing the birds and then measuring dimensions of the beak and the body, and then putting onto their legs 4 bands before we release them so that we can identify them. One is a numbered metal band, and the other 3 are colored plastic bands where the colors are coded to correspond to the number on the metal band. And so when we release the bird and watch it, without further catching the bird, we know the size of the bird and we can relate it to its feeding behavior. The 2 main species on the island are the cactus finch, a specialist on cactus food, but the main one numerically is the medium ground finch, a seed-eating bird that is rather like an ordinary sparrow, about 15 grams in weight. Now, by capturing a lot of birds, hundreds, and watching what they feed on, knowing their measurements, we were able to establish that the bird is the species is a feeding generalist, but within the range of variation, there's some degree of individual specialization, with those birds who are only seen feeding on small seeds, very much smaller in beak size than those feeding on small, medium, and large seeds with this very high beak depth measurement on average up here. So there's variation for forces of selection to work on. Is there evolution occurring now in contemporary time? And if so, what does it tell us about the process of speciation? Well, the first certainly is evolution as a measurable phenomenon. Here I'm showing the array of average beak depth for a whole 40-year period of this species, the medium ground finch. And you'll notice against a background of generally very minor changes from year to year, 2 major events, one here early in the study and one here towards the end, a much stronger event back here. 2 natural selection events with much in common, but because there are some slightly different circumstances, I'll treat them separately. The commonality is a drought and converting that kind of scenery with seed-rich plants in the foreground into one of apparent devastation with extreme scarcity of food, high mortality, and therefore great reduction in population size. In the first episode, the rains failed in early 1977, and 80% of the medium ground finches died. At the beginning, all finches fed on the small, easy-to-handle seeds that were in abundance, but as the supply was depleted by consumption, the birds had to turn increasingly to alternative foods. Principally, this one up here, I illustrated with an icon, tribulus, a woody fruit that requires quite an effort to crack open to get to the seeds on the inside. Only the large beaked birds could do this, so they survived much better than the small beaked birds, and as a result, the average beak size increased until the rains resumed in early 1978, and the pressure on the population was off. So, in short, natural selection had occurred of that magnitude. Now, natural selection is not the same as evolution. Evolution only occurs when natural selection acts on genetically heritable variation. And the evidence for heritable variation is very strong in this particular case, because the average of the mother and the father measurements of beak sizes strongly predict the average of their sons and daughters' beak depth, beak length, and other dimensions. So yes, there is high heritable variation for this trait, and hence, not surprisingly, in the next generation, when the rains resumed and the birds spread, then the offspring were very close to the predicted amount, very close to, in fact, to the parental measurements due to the high heritable variation in the trait. In summary, then, natural selection on a heritable varying trait produced an evolutionary response in a measure of beak depth that we could understand in biomechanical terms when the environment changed. Now, the environment changed a lot after that, and I'm going to illustrate one major effect. So this was 1977 back here. Now, in 1983, there was a record-breaking amount of rain on this arid little island, the largest amount of rain in the last 400 years, according to Coral Core data. Briefly, the rain, the abundant rain, converted an island landscape, which is dry in the dry season and greener in a normal wet season, into a lush, almost alpine meadow of grasses and vines. Vines grew everywhere as the rains continued for 8 months, covering indeed all of the bushes where the finches bred. The most significant factor arising from this heavy rainfall was the arrival of a new species on the island, Magnorostris, the large ground finch. 2 females and 3 males stayed to breed. They were vagrants to the island. They established a breeding population, and gradually the population built up. And so by 2004, at the next time of a very serious drought, their numbers are very large, on the order of about 150 to 200. Well, you can see by their large beaks, and here the lowest one has a tribulus fruit in it, that they must be extremely efficient at dealing with this food. And the summary statement of the events is this, that this species, Magnorostris, outcompeted the larger members of the medium groundfinch population for a diminishing supply of tribulus fruits when food supply in general was going down. Forties then were at a selective disadvantage. They didn't all die, but they were at a selective disadvantage because the rate of mortality was much higher than it was in the small members of the same population, with the result that the average the average size of the beak declined, and the difference between Fortis and Magnorostris in beak size became larger. The drop in average size was very strong. And so this is an example of character displacement that I mentioned at the beginning when I said that when 2 populations come together after previously being separated, they diverge as a result of natural selection and minimizing competition for food. We know now a lot about this aspect of the biology of the finches, the link between phenotypic variation and fitness variation. Now, how about genotypic variation that powers the phenotypic variation over and above what we know about inheritance through heritability studies? What are the genes involved? Now, we have collaborated with a molecular geneticist at Uppsala University in Sweden, Dr. Leif Andersson, who has, and his group have, sequenced the full genome of many individuals of our study population, and indeed all of the species in the whole of the radiation. And by comparing pairs of contrasting phenotypes, they were able to identify genes of significance responsible for the differences in phenotype. So you could think of this as two genomes lined up together. What are they? In this case, large compared with small birds, large compared with medium, and medium compared with small. And by running along the line of this paired comparison, you can see that at one locus here, and in every comparison, there is a major difference genetically between the phenotypes being compared. One of these genes, HMG2, is known to vary in association with body size in cats and in dogs and in rats and in mice and in us humans. In Darwin's finches, interestingly, the variation in genotypes at this locus are associated with variation more in beak size than in body size. So the LL, standing for the large, is associated with large size. The SS genotype is associated with small size, and heterozygotes with intermediate size. And more interesting than that is that in the character displacement episode that I've described, survival of finches varied as in relation to genotype over here, as well as in phenotype. So the LL genotype carriers had very low survival, the SS had very high survival, and heterozygotes were intermediate. To bring you right up to date, the group have done an analysis of beak size variation and found that 6 loci only, independently affecting beak size and beak shape, can account for a remarkable 50% of the variation in the morphological traits. GO3 is our friend, HMGA2, and the other one of significance here, GO7, is a beak shape factor. So in conclusion, natural selection occurs when the environment changes, as we've been able to document and understand. Other species determine the direction of evolution. In our case, it's up or down. And both of these are core ingredients of the Darwinian or classical model of speciation, starting in allopatry and being completed in sympatry. But I haven't said anything about how the species keep apart reproductively. And for that, I'm going to hand over to Rosemary.
So what I'm going to talk about are the causes and evolutionary consequences of rare episodic gene flow between the species. And then one of the most exciting parts of our study, completely unexpected, was the formation of a new lineage, which we've been able to follow from its inception right up through 6 or more generations. But first of all, just because I know you're going to ask me this, if I don't say, we follow the biological species concept, which is that species are a reproductively cohesive group of organisms that are diagnosably distinct. And the reproductive barriers between them are very rarely penetrated, but they are very occasionally penetrated. So what happens when they are? So amongst the finches that we've been talking about, we can't use, but if we want to know what the pre-mating reproductive barriers are, we can't use plumage because males are black in all of them and females are brown. But what we can notice very strikingly is that they differ markedly in size from a 12-gram fuliginosa, small ground finch, up to the 32-gram Magnerostrus, large ground finch. They also differ in beak shape, from the pointed beak in scandens to the blunter beaks in Magnerostrus. And they differ markedly in song. And it turns out that song is really important. So what do we know about song? Well, from work done by Bowman and then followed up by us, we know that song is learnt very early in life during a short sensitive period, which is between 10 and 40 days approximately after hatching. Once it is learnt, the song is retained for life. We know this because we record the birds every year. And as adults, they pair according to their species song and morphology learnt early in life. So a typical sexual imprinting based on learning. So because this pre-mating barrier is based on learning, it is vulnerable, and so it can be breached or broken if a young bird hears and learns the song of another species during its short sensitive period. And this happens about 1 to 2% of the time. It happens for two main reasons. One is nest takeover. If we think of a scandens, a cactus finch taking over a fortis nest, normally it will turf out all the eggs, but occasionally an egg is left behind and it grows up as a fortis in a scandens nest and learns a scandens song. It also happens with the death of the father. Then the mother has to rear the offspring. Females don't sing. So if there is a neighboring bird singing, it will learn that song. And if that neighbor is another species, it will learn another species' song. And we found that when the size difference between the species is small, as it is between a 22-gram scandens approximately and a 17-gram fortis, then when the size difference is small like this, learning another species' song can lead to hybridization. So when we first arrived on the island in 1973, there were two main birds, a medium ground finch, fortis and scandens, were very different in beak shape, and they were kept apart. So the populations looked like this. Then after the El Nino event of 1983, which Peter told you about, the biggest in 400 years, then we began to get hybridization between the two, and we got convergence both genetically and morphologically between fortis and scandens. So why did this, excuse me, why did this integration occur after 1983? Well, in 1983, the island was covered with vegetation. The cactus bushes were covered with vines like this one. And even after the 83 event, immediately afterwards, the island looked like this. So it's not surprising that the Puntia was, many of them were completely destroyed. They either fell over because they had taken up too much water, or they'd been covered in vines and couldn't photosynthesize. So what happened afterwards, that there were very few flowers and fruits formed, and flowers and fruits are the main things that scandens relies on. So the population of scandens, the cactus finch, declined, and also the sex ratio became very skewed in favor of males. So at one time on the island, we had just over 100 males, but only 16 females. And the reason for this skew is that the males are bigger and they outcompeted the females for the few flowers and fruits that were available. So what happened then is that with these few hybrids about, the scandens males were desperate for females when the breeding season came around. And they bred, first of all, with their own females, but then with any female hybrid that occurred. Whereas the male hybrids were outcompeted both for territories and for mates. So we had integration, which was mainly from female fortis into the scandens population. And this went on for years after this event. And the average beak pointedness of scandens became blunter and blunter and blunter over the years and much more fortis-like. So what we really wanted to know is, are there specific genes, alleles, that could have contributed to these changes of beak shape? And so we sent all our blood samples again over to Leif Andersson's lab in Uppsala, and they came across, by putting all the sharp beak species together with the blunt beak species, they found this intriguing region that has the gene ALX1. Now, this was super exciting because we knew that a mutation in ALX1 is largely responsible for cleft palate in humans. So we argued it had to have something to do with the beak cranial facial morphology. And sure enough, it did, because in situ hybridization using ALX1 as probe showed that the expression occurred in the beak region at the time of the developmental process. So what our Uppsala friends did, they took all our blood samples, and they were able to track this ALX gene, one gene which encodes a transcription factor. They were able to track the blunt form moving into the scandens population through this hybridization and back crossing, particularly from the hybrid females into scandens. So very excitingly, we could follow this integration all the way through, which explained why scandens beak shape was becoming blunter. And not only that, but you can actually see it, because these 3 birds were taken out of the net in 2012. The one at the top has a sharp pointed beak, which is as scandens was before introgression, and it is homozygous for the ALX1 pointed allele, whereas these are heterozygous for the pointed blunt allele, so they have the blunt allele and then blunter and more fortis-like. So we excitingly wrote a mosaic genome arising from hybridization between 2 species generates phenotypic novelties that would be under selection in a new environment. An increased genetic and phenotypic variation could fuel a rapid change along a different trajectory. But we never thought we'd actually see it, but amazingly we did. And it happened in this way. A bird arrived on an island at the time when we had all of the individuals banded on the island. It looked like a big fortis, a really big fortis, because fortis is 17 grams, this is 28.5 grams. It was a young bird when it arrived, and when it, the next year, when it started to sing, it had a completely different song. And we banded it 5110. Well, later, by taking the blood samples, we found that it had come, all the way from Hispaniola in the south. It was a Conirostris on Hispaniola, and somehow, we don't know how, it had got to Daphne. It was a massive bird. It got one of the best territories on the island. It sang, defended its territory, but it couldn't get a mate. After all, it had all the wrong, it was all the wrong size, all the wrong shape, and it had all the wrong song. And it tried for 2 years. And then at the very end of the second breeding season, right at the end, a female fortis had lost her mate had died, and she moved into this beautiful territory and mated with 5110. They produced a number of offspring, and then in the 2.5-year drought with the character displacement event that Peter told you about, all these died, went out, leaving these 2, a brother and sister, highly inbred. So after 2.5 years, the brother and sister mated with each other, produced 26 offspring, all of 9, except 9 survived. We had a daughter breeding with her father, a son breeding with a mother, and the rest of the sibs breeding with each other to produce the next generation, All these birds, we had blood samples from them all, showed genetic transmission from 5110, all sang 5110's song, and all were large like 5110. But what was extraordinary was that it had been an allometric shift. So this is the Fortis population. This is the Conirostris from Hispaniola. And what we call the big bird lineage was lying here, not intermediate, but it had a relatively large beak for its body size. So we know that Peter has introduced you to HMGA2, which was body size. I've introduced you to ALX1. They occur on the same chromosome, closely linked, and we know so far that recombination between them has caused this body size shift. But we're looking into this further at the moment. What we do know is that these big birds love tribulus seeds. So we wanted to know how efficient they were at cracking them. We measured the cracking times and found out they were just as efficient as Magnorostris. So if there's another drought, tribulus is left behind, they're likely to do very well because they are as efficient as Magnorostris at dealing with them, but having a smaller body, they require less food. So this new lineage is behaving as a separate species, much larger than its nearest relative, Fortis. It differs in size from the other birds on the island. So this is Fortis, here's Magnorostris. This is the gap where Magnorostris outcompeted the large Fortis, which were lying here, and the big birds have moved right in. They differ in song. This is 3 generations of the big bird song, very different from Fortis song, Scandin song, and Magnorostris song. They hold contiguous territories, which they defend against each other, and these territories overlap with Fortis, Scandins, and Magnorostris. The blue dots are the territories before the drought, the red dots after the drought, and the more red dots over the cliff. So thus the new lineage is functioning as a separate species. Will it die out through inbreeding depression? It might, but it hasn't yet. Will the genetic variation be augmented through genetic gene exchange, but learning the wrong song? It might, but as far as we know, it hasn't yet. Whether it survives or not, this new lineage gives insight into how a new species could arise and either persist or become extinct. So in summary, when we first arrived, there were 2 species on the island, Fortis, Scandins, clearly distinct. After the El Nino of 1983, they began to fuse. Magnorostris came in, outcompeted the largest Fortis, and the big birds moved right in. So how important is introgression as a source of variation for adaptation in new environments? You'd be surprised to know that when we first sent a paper suggesting this many, many years ago for publication, it was turned right back with birds don't hybridize. Of course, now we know that birds do hybridize. We know that hybridization or introgression, genetic introgression, occurs from viruses through bacteria in many plants and in all taxes of animals. Even in our own ancestry, Svante Pääbo has shown that genes from Denisovans and Neanderthals integressed with our own modern human lineage. And we know that introgression happens also in many diseases as it becomes important, including cancer. So we thank our many graduates, postdoctoral fellows, collaborators. None of this could have been done without them. The Galapagos National Park and Darwin Research Station for logistical support, and our funding agencies. So I want to just leave you with 2 messages. We live in exciting times. The genomic data are rapidly accumulating. This considerably enhances our field studies, but reciprocally, and I want to emphasize this, a reliable interpretation of genetic data requires an understanding of ecology, evolution, and behavior in nature. It's not good enough to just go out collecting the genes. Secondly, it would be this would be a conservation message. This wonderful NASA picture of our globe captures the uniqueness and fragility of our planet, with biodiversity being its defining feature. We've shown you that environments and populations are dynamic, they're constantly changing, and for a healthy environment, we must keep them both capable of further natural change. Thank you very much. So,
I think we have to click on now.
Thank you very much, Dr. Peter and Rosemary Grant, for this wonderful talk. We invite now to our audience to write their comments and questions in the chat panel. Liliana will be ready to read your questions and your comments. And we hope you have very exciting and wonderful thoughts while you were hearing the talk. So let's start with the comments. Liliana, thank you very much for helping us with that.
My pleasure. We have some questions in the box. And Gonzalo Fajardo asked, in your mind, after more than 40 years of studies, are evolutionary process in finches still unpredictable?
Sorry, is that...
I can understand that one. I just say that remarkably, I don't hear very well, but I heard that question, and Rosemary hears everything. and missed a key word there. But no, on the predictability of evolution, it's clear that in the short run, if there is a large amount of genetic variation, evolution is predictable, providing one knows the context, that is, the environmental forces or conditions under which selection is likely to occur and evolution to follow. But I would also stress that evolution over the long term is largely unpredictable without knowledge of that environmental fluctuation. You can't predict when the next drought will be, for example, with any precision. But given the drought, then we can predict evolution knowing the genetic variation underlying traits and knowing the ecological relationship between organisms and their environment. In our case, it's a case of beak sizes and food, but you could think of other organisms with different traits having different requirements in their feeding ecology. So in short, in the short term, evolution is predictable under specified circumstances, but the circumstances are often unpredictable.
We might have a volcanic eruption.
Well, yes. Can you predict that?
No.
Should we stop share?
Should we stop share or just keep this up?
Did you want to stop share or?
Stop share if you prefer.
It's okay. I mean, we can leave it like this. It's fine.
No, it's fine. We have another question here by Analia Bender Gordillo. She asks, what was the most challenging part when taking data in Galapagos?
What was the most challenging part of getting data? Actually, the collection of blood and catching the birds was not really very challenging because the birds are so tame. And they didn't seem to mind that we handled them, measured them, took a tiny drop of blood. You only need the minutest part of blood for DNA analysis because the blood in birds is nucleated. So what was the biggest challenge? I think maybe just getting onto the islands and possibly making sure that we took all our water with us because we were staying on the islands for 3 or 4 months at a time. So the logistical things were the most challenging, but actually collecting the data was not too bad.
Yeah.
Thank you. We have another one. Professor Angela Amarillo asks, are there changes in diet of finches caused by the introduction of exotic species or by finches eating on human food remains? If that's so, then how did changes could change the pattern you talk about. Have you noticed any difference among population inhabiting populated versus unpopulated islands?
Yeah, that's a terrific question. We purposely did our work on uninhabited islands and islands so we did Daphne and also Henabesa, islands that have never had any human population on them and have not had any introduced species like goats or anything like that. Whereas things are very different on the inhabited islands, where there has been, particularly islands like Floriana, where there's been a complete turnover due to the introduction of humans. And so, for example, David Steadman's work, when he looked at the fossils that he'd found of finches, one of the most common fossils was Magnorostris. Magnorostris isn't there now. And there's been a whole huge turnover due to the introduction of humans and plants and animals. So that is a huge problem. And also, even on places like Daphne, we're always looking out for and hoping we don't find them of introduced plants that sometimes have been brought over by boobies and things like this.
I would add just a little bit to that. Other people have studied the microbiome of finches around the town of Puerto Ayora on Isla Santa Cruz. They've asked the question whether the microbiome differs between birds living in the urban environment and then birds living off in the bushes, as it were, away from settlements. And they have found differences, and I don't know how profound those differences are, but clearly there is a dietary shift of finches in towns. And as we know, more generally, there is in birds in urban environments throughout populated world, Europe and North America, and so on. And so the question of evolution, though, is an interesting one. I think so far there's no strong indication that the beaks of the finches are being altered. There's little indications that maybe, but I would say that it's an open question still whether settlement or association with human society is going to, in the long run, alter morphological features of the finches through adaptive evolution. That's an open question and a long-term, there'll be a long-term answer eventually.
Thank you very much. We have some comments in the box. Analia Bender, thank you so much. James Montoya Lerma, we enjoy the talk. Congratulations and thank you for sharing the info. Greetings from Univalle, Cali, Colombia. Valentina has a question. I have one question. What could be the implication of this type of hybridization process on conservation strategies? How can we take them into account in the process of decision making?
Yes. Again, I love that question too. Because I think it is a very important question, because in a lot of, I'm not quite sure what in Colombia, but in a lot of places, they want to keep the purity of the species. Whereas actually, by looking back, in almost what has happened in evolution, you find that no species is really pure. And so there's always been the question if a population goes right down to only a few individuals. And actually, I heard a talk just this week where some ferrets in North America had gone right down to a few individuals and they were trying to save it. But actually, there is it came from a European ferret. And the obvious way to get an increase of genetic variation, which would allow the population to survive, would be to actually hybridize these European ones, which was an ancestor, with the North American one, and then it would probably do very well. But there's a whole group of people who want to keep the purity of this ferret because it has little black socks on its legs. But actually, I think when you're weighing up these sorts of things, you've got to weigh up what would input of genetic variation do? Would it save that population? Or is it too far away, too far apart, and would integration actually have deleterious effects? So, but I think it's something that needs to be thought about and realized that an input of genetic variation can sometimes help to save a species that is going extinct. But it's something, it's not a clear-cut yes or no. It's got to be, all the pros and cons have got to be weighed. Mm-hmm.
Thank you. We have another question from Jose. Jose has two questions. First, what do you consider that is the most important component for species radiation? Because we have population with a great genomic radiation, but uniform environment and population with lower genomic radiation, but higher environmental variation. And the second one was already answered.
Okay. So, did you quite?
I'm afraid I didn't hear all of the words of that first question. Something about high genomic variation in with low environmental variation. Was that the question? A lack of correspondence between the amount of genomic variation and environmental variation. I don't know if that was the question and why that is so.
Yes, I think you understand right the question about the relationship between genetic variation and environmental radiation.
Yes, well, there wouldn't be you wouldn't expect an exact correspondence between these two sets of variation, because the third axis of variation, as it were, is time. And through time, environments change, as we know. And so what we see as a relatively uniform environment, let's say a large desert region, through time would have changed from one state to another. We don't know the extent to which the barriers to interbreeding between partly separated populations would have been lower or higher in the past, unless we have detailed information. For the Darwin's finches, all we know, excuse me, is that the islands were changing in their size and configurations over the period of a million years. So environmental variation that we see today across the archipelago is not the same as the amount of variation through time across the same archipelago. There's a very large amount of extra variation. I think of that when I think of studies that are done in continental regions, tropical rainforest studies, for example, desert organism studies, or even in marine environments. And I think that it's good to make the correspondence or to establish the correspondence between organismic diversity and environmental diversity in current time. But unless we have a good understanding of how the current environmental variation arose and whether it's been stable or unstable in the past, we really are missing an important ingredient in trying to understand organismic variation. Complex and very challenging and very interesting, a great challenge, whatever organisms one likes to study.
Thank you. We have a last question from Jacobo Pinto. Hello, thank you deeply for your work and presentation. I want to ask you about the subject of integration and hybridization. How do you think we can study this phenomenon in other ecosystems where the distribution of the population's birds are not limited by an island geography? It's to say, how do you think we can study the effect of integration in, for example, in a tropical forest like in the Amazonia?
Yes, I know that it's tough, but I think what people have done is that they have, first of all, when they have caught the birds, they've taken a little bit of blood from that, and remarkably now with techniques of especially techniques that we were referring to in the talk. They're able to see exactly, to go back and to actually see if there had been integration in the past and with what species. So I think it is possible, even in something as dense as a rainforest, if you have birds that you expect that are very similar, let's say a group of parrots or a group of tanagers. Yeah, tanagers would great one, a group of tanagers, then if you can, when you catch the birds and you take measurements and you take the small drop of blood and then analyze it genetically, then you can see by looking at the phylogeny where, how closely related they are, and whether there had been, the other thing you can use nowadays, there are various techniques which you can use to see if there has been integration in the past. So they're usually called ancestry techniques and things like that. Actually, the same sort of on a different level, you know, this 23andMe for humans, and that sort of thing. You can use ancestry for looking at the relationship between different species of birds and how, whether there's been integration or not. And I think that's a very exciting area now. In fact, it's so exciting and so popular that people are just looking at the genetics, which is why my little thing is that you can't just rely on genetics. You must also know the behavior and then the ecology and of the organism that you're looking at as well. So sometimes it becomes almost easier to do the genetics than to do the hard environmental work, but it's so important. Mm-hmm.
Well, thank you very much, Dr. Peter and Dr. Rosemary Grant, for this exciting and wonderful talk. Thank you for being so kind to us by sharing your research.
I think you have been kind inviting us.
Well, you are very welcome every time you want to come to talk with us, and we are very grateful with you. For everybody else, I thank you for being in touch with the doctoral program in environmental and rural studies. Please stay tuned for our next talk next month about marine water environments. We'll have a new invitation for you for the end of October. So